Okay. So, first thing, yesterday's some question was there yesterday with about the monocystronic question. Correct? There was some cystronic, polycystronic, monocystronic. I think the answer yesterday, the website answer is not right. I think it was not right answer. Basically, when we talk about monocystronic or polycystronic, what does it mean is that if a single mRNA codes for a single protein, that is going to be monocystronic. So in that case, most of the mammalian genes are monocystronic, correct? Of course, there are some exceptions there, wherein you have the different small peptide is ahead of the actual protein coding region. Those things are there. But in bacteria, many a times you will see one single mRNA coding for multiple proteins with start codon and stop codon different for each protein. Okay, so when we talk about, when we say the polycystronic or monocystronic, what does it mean by is that uh, insert, okay, saves. Okay, so this is one ORF, correct? And then same mRNA has another ORF downstream. Okay, that would be the stuff that when we talk about polycystronic or monocystronic. Here is a start codon here, and there is a translational stop codon here. And next downstream in the same mRNA, if there is a start codon there and followed by a translation termination downstream, that is polycystronic. But if the mRNA has only kind of only one uh, coding region, just like this, one stop codon, one start codon, and one stop codon, that would be the monocystronic. So many of the mammalian genes are monocystronic, and polycystronic you will see most often in prokaryotes, correct? And we talked yesterday about viruses, correct? Viruses are generally polycystronic. But one speciality of viruses is that generally, many a times, including HIV, what it happens is that it is kind of a monocystronic. It is translated single long protein, and the proteins are separated. Different proteins arise after proteolytic cleavage of the long protein. So in this case, if this is a single operating frame, still I think it will be called as monocystronic by definition, because you have one ORF. Okay, yeah. So that will be the thing, clarification. And I have updated yesterday's slides. You can have a look. And I wouldn't recommend the first question link yesterday we discussed about, because a few of the concepts I'm not very happy about the concepts. The answers that was given, it was not very great. So I don't recommend that first link. Okay, and I have put a comment there. So to continue yesterday's thing, what we were talking about was, I think we were here, correct? So replica plating and disruptive cloning. This was part of the PBR 322 plasmid. That is one of the first generation cloning plasmid, which has both ampicillin and tetracycline markers. And by cloning something at the BAM1 site, which is inside tetracycline, selection marker would disrupt the tetracycline resistance gene and would give rise to sensitivity to tetracycline. Okay, so here you can see here when you select on tetracycline plate, only few of them are going to grow up and the rest is going to be uh, not growing. And depending on whether it grows or not, you can decide whether there is insert there inside the system. Now going ahead, the cloning vector was more of just cloning a fragment of the DNA from any source, whether it can be a genomic DNA or whether it can be a cDNA library that we discussed yesterday quite extensively. We are not looking forward in that case to uh, these vectors doesn't contribute in the expression of the genes which has been cloned there. So that's why it's just generating multiple copies of it. But if you generate large fragments of genomic DNA and put in the same organism, like E. coli genomic library, if it is a large one, and then you put in E. coli, it is likely to be recognized wherever there is a promoter region that is going to be recognized, transcribed, and then translated in that case. So you still may get proteins out of it, but generally native, most of the native promoters are not very strong. So if you want to get expression of a protein in a heterologous system, what do you mean by heterologous system? Heterologous system means you are getting a protein of another organism in another organism. For example, if you are expressing human proteins in E. coli. E. coli is a heterologous system for the human proteins, correct? So to express human proteins or cerevisiae proteins in E. coli for the purpose of bulk production of proteins, what do we need? So if we look into uh, the if we look into what all elements are required for protein expression, we'll discuss in the next slide. Before going into that, I would like to discuss this vector. This is the PUC vector. PUC 18 is there, PUC 19 is there, depending on the selectable marker. Here in this case, the beauty of this system of plasmids is that you have something called leg C control region or leg I promoter region. Okay, this drives the expression of a downstream gene that is leg C gene, which what does it do? It codes for a beta galactosidase gene. So why it is important is that if you use some of the markers, so here, what is the importance here is that many of these vector systems have multiple cloning site. That means a number of multiples of restriction enzyme sites have been put into so that you can choose which uh, enzyme fragments or compatible ends you get it. If you want to clone in BIMH1, you have a site for BIMH1, compatible ends will be generated. If you want to use ECOR1, you will get it. If you want to use PSC1, you will get it. So multiple cloning site is nothing, it's a part of a plasmid wherein you have multiple restriction enzyme sites are there so that you can choose your restriction enzyme of interest. Now coming back, so what is important here is that these multiple cloning site is ahead of these beta galactosidase gene. The importance is that if you insert something at this multiple cloning site, it is going to, most of the time, it is going to disrupt the translation or transcription of these, mostly the translation of these beta galactosidase gene. So the clones which are positive will disrupt the function of galactosidase, beta galactosidase gene. What is the importance here is that when you put in IPTZ, okay, and uh, if you induce it and select on X-gal plate, what you can see is that if there is no clone, beta galactosidase gene is going to be active, and it is going to give rise to a blue color, blue colored colonies. Okay, so this will help us in visual selection of the colonies. If it is blue in color, you don't want to pick it up because there is no insert in that vector. Okay, likewise, when something is cloned in the beta in the multiple cloning site, what it will happen, it will disrupt the beta galactosidase activity, and it will yield, in the presence of XL, you will get white colored colonies in the case of E. coli. So what does it mean? You can, white ones have insert inside, and blue ones doesn't have any insert in your vector. So you can do blue white visual selection. So this is very quite popular in the cloning strategies. So I just wanted to discuss this one. So next, what we were talking about, expressing human protein or cerebracea protein in E. coli. Then what do we need? Okay. So those things will come before going into that. I think there are other vector systems. So far what we have been discussing is the plasmid cloning system, correct? It's a autonomously replicating sequence DNA element, and these are called episomes, correct? So these episomic, these are plasmids are there. Next, there are, in the cloning strategies, there can be other than the plasmid, you can have viral vectors which you can utilize. For example, lambda bacteriophage is widely used for uh, cloning purposes. The main purpose here is that, as you all know from your, I think some of the introductory microbiology courses that we talk about, the bacteriophages will have lytic or lysogenic pathway, correct? So lytic means it will be maintained as an episome and lead to the lysis of the host cell, and it comes out. In the case of lysogenic pathway, the episomal genome of the virus is integrated into the genome of the host bacteria and integrates in the genome, and it is maintained. Now, depending on the nutrient status, you can switch to the lysogenic and switch to lytic and comes out of it. Okay, so most of the time, I think when there is a nutrient enriched condition, then it goes into lytic phase. So these plasmids or the virus which are coming in the lytic phase can be utilized for cloning purposes. Okay, you can clone and then replicate inside the host and multiply inside. So you can utilize the bacteriophage for cloning purposes and do a lot of genetics. Then you have the yeast artificial chromosomes. What you do is generally is maintain as a big plasmid, the parent plasmid before the cloning of your insert is generally maintained as a circular plasmid. Then what you do is you clone in the multiple cloning site here also, you clone the big fragment, and you later on, after cloning the insert, you linearize in such a way that it behaves like a chromosome in cerevisia or in the yeast of interest. Okay, so as we talked yesterday, that these artificial chromosomes can take up huge chunk of or much larger size insert in the artificial chromosomes. That is the main advantage. So it helps in studying um mammalian genes, or you can generate large mammalian genome fragment and then use for this function. For example, the first genome like cell division proteins, many of them were discovered by putting genomic library of human in yeast vectors and introduced into yeast, and it complemented the defect in the cerevisia. That's how many of the cell division genes were discovered. Okay, so there is importance for these kind of studies. Again, next, this is one of the plant. I just wanted to include one plant vector. So one is the TI plasmid, which is kind of utilized for tDNA insertion lines. So you have certain something called tDNA insert stuff. So TI plasmid is generally used for many of the Arabidopsis studies. It behaves pretty much the same like the mammalian plasmid, which we use, wherein the cloning is done in E. coli, then finally you put into the mammalian system. So TI plasmid or tDNA insertion lines you can get in many of the plant studies. Then for humans, one of the viral vectors that we very commonly use is the lentiviral system. Lentiviral system is derived from the HIV virus. So basically what it does is that you package and generally you have a plasmid, you multiply and then package into lentiviral vectors as a virus, and that enters the host cell. After entering the cell, what it does is that, because the genome of lentivirus is RNA, correct, single stranded double-stranded RNA, and that no single-stranded RNA genome, and that goes and through reverse transcription you get the double-stranded, single-stranded first, and then from the DNA polymerase you get the double-stranded DNA. This
Sir,
double-stranded DNA, yeah,
when you're using a TI plasmid vector,
yes.
how do you get past the cell wall of plant cells?
Generally, they introduce either the this thing infectious. I think they use one of the
Or like a
uh,
tobacco mosaic viruses?
yes, yeah, like that
Okay,
you can use, or electroporation is also utilized. Yeah,
okay.
so uh, so in the case of lentivirus, once it is inside and converted into double-stranded DNA, this can go into the nucleus and integrate into the genome. So this integration is random. There is no specific site wherein things, the viral genomes are integrated. So the one of the very important property of lentiviruses is that it infects even non-dividing cells or less actively dividing cells. So why it is important is that many of the viral vectors that is used for studying human genes or infecting or transducing human cells, many of them prefer to infect dividing cells. So that limits, because many of our cells like neurons or cardiac tissues, all this doesn't divide much. So then the efficiency of introducing or transgene or your gene of interest inside those cells will be very much limited. But lentivirus doesn't have much differentiation between dividing, non-dividing. So then lentivirus becomes very, very important in that aspect. Another one which is very, very hot right now is the Okay. Before going into that, the selection markers, we talk about selection markers in E. coli yesterday. You have tetracycline selectable marker, you have ampicillin, you have spectinomycin, you have chloramphenicol, so many markers. Many of them doesn't work in mammalian system. So for mammalian system, the more popular selection markers are blastocidin. Very popular is the puromycin because it is a very good selectable marker. That means wherever that transgene is there, it gets selected. But other markers are utilized, but not so efficient because it doesn't select. There is the selectable power for these antibiotics or these chemicals are not so high. So there will be lot of cells when you treat with blastocidin. It doesn't. It allows the growth of those cells even if they don't have the plasmid. Okay, so selection efficiency is not so high for these other markers. Of course, people always use, we also use blastocidin, all these things, and but one of the most popular is the puromycin. Puromycin, basically it targets at the elongation step of translation, and it is quite good selection marker for mammalian cell lines or mammalian or high eukaryotes. Another many times people use fluorescent markers as well for selection of the plasmid, because when you have fluorescence, you can put into fluorescence activated cell sorting FACS technology. You can select those cells which are positive for that particular fluorescence marker. So even fluorescence markers are quite popular in the eukaryotes or mammalian system. And one more very interesting vector, that viral vectors that is coming up is the adeno-associated virus. Okay, adeno-associated virus are quite it's a small genome or small size virus. Generally, it has two proteins, RAP and CAP. And, red and RAP and CAP, these two compasses around 5 kV in size. So the viral genome is just around 5 kV, and it requires adenoviral proteins, something like the E4, E2A, VA, E1, uh, uh, the adenoviral proteins E4, E2A, and VA for its replication inside the cell. Okay, this adeno-associated virus. Another one more requirement is that E1, okay, E1 plus HEC293. E1 plus is kind of a receptor for adenoviruses, because if you have to put in together adenovirus as well as the adeno-associated virus, this receptors requires for the entry of the viruses. So the cell line should have that receptor E1 plus marker for the entry of AAV gene. Now, what is interesting about this AAV is that it doesn't, most of the time, doesn't uh, integrate, but sometimes it does integrate. If in the presence of RAP protein, the AAV gene can integrate in chromosome 19 of human cells, okay, in chromosome 19. But if you don't have this RAP protein, what it happens is it doesn't integrate at that site, so it is maintained as a episome. Why it is very hot now, AAV, adeno-associated viruses, is that there is a tropism. What do you mean by tropism? Tropism means it doesn't, some of the AAVs doesn't infect or any type of cells, but it has a very uh, limited limited number of cells it can infect. For example, some of the AAVs can infect only like neuronal cells more efficiently. rather than epithelial cells. Then it helps you for a specific targeting delivery of transgene, which you can put uh, in the AAV. So what people have done is there are inverted terminal repeats, ITRs are there. Okay, in between that, the gene of your interest can be put. For example, some of the uh, genes which are important for or which is lacking in certain disease condition, you can put it here and allow it to express in the particular tissue. That is the but so after putting to get it packaged, what you have to do is you have to put the transient instead of wrap and cap, but wrap and cap is put in a separate plasmid. Okay, so that this can be packaged in the AAV viral particles. Okay, so by replacing the wrap and cap, you generate this kind of construct which is called transfer plasmid. This construct is what is going to be packaged inside the AAV viral particles, and it packs only a single strand of the DNA. It's a single stranded DNA virus. So in the presence of RAP, CAP, and adenoviruses, that is called the helper plasmid, in the presence of these factors, these construct, which is flanked by ITRs, will be packaged in the adeno-associated particles, which can be used for introducing your gene of interest, cDNA or shRNA in this case, into a particular tissue. For example, neuronal tissues, which is very difficult to be transfected by many or other viruses. So, and when after packaging, you don't have wrap or cap, so it doesn't integrate into the genome, but it allows the expression of your transgene. So it has become very, very popular in the recent times because of its because it can be utilized for specific tissue delivery of transgenes of your interest. So
Sir,
yeah.
in over here, since it won't get integrated into the DNA,
Hmm.
it won't be transferred to the next generation of the HK cells, right?
No, it's not. It can be lost. That's why if you are treating with AAV, then you have to administer uh, uh, at regular intervals. For example, if you are treating someone, then you have to be treating at regular intervals. That is, of course, because it doesn't integrate. So what it helps is that it doesn't disrupt any genomic organization, so it is safeguarding the genome. So if you think about the safety aspects of your way of delivering your transgene, this is little bit safer. That is the that's why it is becoming very, very hot. Yeah. And tissue specificity, correct, because there are the other things. People have utilized lentivirus or adenovirus for kind of gene therapy purposes, and they had a lot of problems, especially adenovirus. I think there is a story which was no lentivirus, I think, for treatment of a child in US, and somehow the child died very soon because of the genomic disruption. So there are issues, safety issues for other integrative viruses. And I think so far no one allows any integrative vectors for germline modification in humans. Germline means that will be transferred into the next generation. Okay, that is not approved yet. Yeah. So then we have been talking about expressing some genes in the mammalian cells or in the bacterial cells for large amount of protein production. What has been seen is that, of course, this has been utilized, and many proteins, including like hepatitis V vaccines, then you have cytokines or insulin has been produced in uh, uh, the clone and express and utilized for various purposes. So now before, if you want to express a protein of your interest in, for example, in prokaryotic eh, vector, expression vector, what all elements you require it. That's very, very important to look at. Okay. So here, of course, every vectors, plasmid vectors or other vectors, you need to have a origin of replication. Here we are talking about plasmid. Origin of replication is required. For our convenience, we need to have a resistance marker. Most of the time we are using antibiotic resistance marker. And then you have the restriction enzyme sites. Okay, so the main components you need to have is you need to have a promoter. So this is a prokaryotic, you have -10 and -35. Okay, in the case of eukaryotes, you need to have data box, correct, and the -35 equivalent that has to be there. So you need to have an active promoter so that there is firing of transcription. Is the transcription firing is enough from the plasmid? No, correct. After transcription, the mRNA has to be translated. So to do that, you need to have in the mRNA, you are getting it, that should be a very efficient ribosome binding site, or we talk about Sine-Dalgarno sequence, correct? So those ribosome binding sites has to be there after the transcriptional start site. This should be in the mRNA you are getting in. So you need to have ribosome binding site, then followed by your ORF, that is the coding region of the open reading frame of your protein of your interest. Coding region of the protein of your interest has to be there. And in that coding region, what we need to have, you need to have a translation start site, then you have a coding region, then translational stop codon. Okay, is that enough? No, correct. You need to have the transcriptional terminators. So if you look at it, you need to have a promoter, then you need to have a ribosomal binding site, then open reading frames which have start and stop codon for the translation to be starting and terminating, and you want it to be saving energy and proper transcription to be terminating downstream. In the case of eukaryotes, both of them is applicable. Of course, the sequences between each organism of your interest is going to be different. If it is a human or the mammalian or any cerevisiae, you need to find the right promoter. Then if it is going to be translated, you need to have a ribosomal binding site, and you need to have this coding region. And in the case of eukaryotes, what we need is poly A signal, correct? You need to have signals of polyadenylation, and then you need to have a transcriptional terminator. So all these elements have to be there in your construct so that you can express your gene of interest in prokaryotes or eukaryotes. So far, in the last few classes, we have been talking about non-coding RNA, correct? In that case, you need to know ribosomal binding site is not important in that case because we are not looking for ribosomes to come and translate that RNA. So then what is important is the sequences which are flanking that mature, for example, microRNA expression. You can't be just putting in the sequence of the microRNA and transcribe it, and it won't function because the processing that we talked about earlier, DROSA was there, then DICER, all these recognize the features or the sequences which are lying in that primary transcript, correct? So all those features sequence, like the for shRNA you need to have a loop structure. Even microRNA, we look at it, it has a loop structure in the primary transcript, correct, which is recognized by the DROSA, and the RNA binding proteins can come and start processing. So you need to put in all those elements so that it is properly processed, it is exported out of the nucleus, and then it matures and it goes into risk complex and then go for its activity. Likewise, like tRNA or RNA, you have to express with those flanking sequences so that it is processed properly and matures properly. Otherwise, the system will just recognize as an aberrant RNA, and RNases will come and degrade it, and there won't be any function or any activity of those RNAs. Okay, so you need to have these are genetic elements which are to be there in a expression vector. So if you want to express a protein, all these you have to keep in mind, and then you have to look at how much distance the ribosome binding site is from the start codon, because that distance is also important, correct? Just sequence somewhere, putting somewhere here doesn't help the purpose. After binding, it should be at the right context with the start codon, the ATG, so that it is much efficiently translated. So all those have to be kept in mind when you are uh, cloning something for protein expression. So now to go ahead, once you clone into expression vector, next step is generally you want it to be, you want to purify. So one of the examples that people use is to make easier purification. What it does is that people put in extra amino acids. Okay, so what do you mean by that? You have a protein of interest, which is okay. It will be fine, I think. So this protein, the blue circle, here is your protein of interest, and here you are putting extra sequences. In this example, is the seven histidine sequences, correct? Seven histidine uh, extras. These are called tags, okay? Histidine tags. This is called histidine tag. You will have GST tag, you have simulation tag, so many tags, or GFP, whatever you are putting in other than the gene of your protein of your interest, that is the tag. It can be histidine tag, it can be GST tag, it can be GFP tag, it can be anything. Okay, this tag in this example, histidine is generally, if you look at it, it's generally what it should be, negatively charged. Okay, it's negatively charged at neutral pH. Okay, so that neutral pH, at neutral pH, it's going to be negatively charged, so it can bind to cations very easily. Generally is the divalent cation can bind very easily. So when you use histidine tag, what it does is that there is a resin immobile phase of a column, and that has coordinated cations, so that when the histidine comes in, it binds to that divalent cation. Then after some time, you can rip off the divalent, or you can put in something which competes with histidine, so that the histidine tag proteins comes out. Okay, that is one thing. Or many a times what it does is people put certain proteolytic cleavage site between the protein of interest and the tag you are putting in this case histidine tag, correct? So after, for example, TEV, uh, there are endonucleatic proteins. So your proteolytic cleavage can be generated at specific sites between these protein and histidine, and by the treatment of that protease, you can release the protein of interest. Then it's much easier to purify your protein of interest. Otherwise, you have to search around like multiple columns, then multiple ways to get the pure protein, correct? So this kind of tagging helps in better purification. One thing which is very, very important for this kind of purification purposes is the to know the PI of PI, isoelectric point. You must have heard, correct? So isoelectric point of the protein is very, very important. So in this case, we are talking about just a few examples. Single amino acid. You have an amino group and carboxylic group. Right. This carboxylic group and amino group, depending on the pH of the solution in which this amino acid is, it can be protonated or deprotonated. Deprotonated. So in the case of normal case, generally acidic ones becomes deprotonated when you increase the pH. Okay, when you keep on increasing the acidic carboxylic acid group is deprotonated first, then the ammonia group, which is more basic, is deprotonated later. So the depending on the pH, you can have different charges on the amino acid. It can be positively charged, it can be neutral, or it can be negatively charged. So depending on the pH, you have to maintain. Now, how it is important is that the Ka, dissociation constant, acid dissociation constant, pKa, the pH at which the acid, here you have two acids. For example, this is aspartic acid, one of the amino acid. So in this case, you have two two carboxylic acid and one amino group. So at pH, once it reaches to below pH of 2.1, all are protonated and it is one positive charge. But above 2.1 and below 3.9 pH, what you are getting at is this one is protonated, this one is deprotonated, so it becomes neutral pH, that is called the deuter ion. Okay, and then above 3.9 and below 9.8, both acids are deprotonated, so it gives one negative charge. Above 9.8, you get two negative charge. Why this is important is that depending on the pH of the solution in which your protein of interest, if you put in, that will have its own charge. For example, if you have a polyaspartate protein, for example, if you have polyaspartate, what will happen? If you are putting at 7 pH, then it is going to be negatively charged. So you can utilize, suppose if at pH 7, you can use something like cations or positively charged resin so that you can purify that protein of interest, which is polyaspartate. But if you put at pH below 2.1 and try to use the same column with of cation column, that won't work because this has itself positively charged, correct? So the our below pH 2.1, it's not going to work. But if you put above pH 9.8, this doubly negative charge, so the binding efficiency is going to be much better as compared to pH 7. So the charges on the amino acid contributes to define the PI of a protein. So if you have polyaspartate, is going to be negatively charged at pH 7, and it will be highly negatively charged when you put a pH above 9.8. So you can choose the column you are going to use when according to the pH of the protein. So these strategies are very much utilized. For example, in certain cases, what people do is you put, for example, polyaspartate, you put at pH 7 first, then utilize a positively charged column to purify once, and then shift the pH of the solution into below 2.1, for example, and use a natively charge. So that will get rid of impurities which are supposedly not behaving in a similar way. So you can purify the protein of your interest. So generally for pi of a residue, it is average of the pKa's of two of the charges. In this case, the pKa's generally you calculate around the first 2.1 and 3.9. So anything in between, the charge is going to be same on the amino acid. So acidic ones, you take the first two pKa's to calculate pi of that amino acid. In contrast, when you are using like lysine, another amino acid, wherein if you look at it, you have two amino groups here, which can be deprotonated at two steps. So to calculate a pi of lysine, you will take the last two pi pKa values and take the average of it. For example, here pKa 3 and pKa 2 will be taken and take the average of the two. That will give you the pi of lysine. So after calculating pi for each of the amino acid in a long protein, you can calculate the pi of the full length protein, how it is going to be charged or how it is going to behave in a particular pH in a particular solution. So this can be utilized for purification purposes of the proteins. So I think we are not going too much in detail about this protein purification. What I would like to highlight is that in a vector for protein expression, what you require is a promoter. You need to have, if it is going to be translated, there should be sequences which allows ribosome to come in properly and translate it. So then if it is then you need to have a coding frame which have a start codon, stop codon, then transcriptional termination. So now the question is, in many of the cases, you want to express two proteins, or if you want to generate polycistronic construct, how will you do it? For mammalian expression, there are certain things called internal ribosome entry site. We talked about, I think, in earlier class. Internal ribosome entry site doesn't require ribosome to recognize methylated guanosine cap. Correct. In the mammalian translation, you need M7G cap to be recognized by the translational apparatus. Correct. Then only it look for the start codon and get translated. If you want to generate a polycistronic construct, what people can do is, after the first codon, you insert a ribosome entry site IRES sequence here, and you can put second ORF there that can be gone into a eukaryotic system and translated into protein expression. Hello. Can you hear people?
Yes, sir.
Yes, sir.
Okay. In fact, power went off. That's why it was gone. So anyway, um today's class is over with that slide. But what I wanted to point out is that to express a protein, what all elements in the uh, plasmid is required. That is what I was discussing about. And then how to clone we have discussed earlier, correct? So pretty much is covered. So in the next week we'll have two more classes, and then we wind up the course. And for today I think there's much. And, do you people have any questions? Any questions, guys? Okay, then see you in the next class.
Sir, question.
Yeah, tell me.
Using a vector, is it possible to insert all the sequences that are required for a ribosomal formation? Maybe all the sequences required for an rRNA, or is it too large?
No, it can be, it can be. It can be done. Mm.
Oh,
It
okay.
can be done. That people have done. You can do.
Thank you, sir.
Any more questions? Then see you guys then in the next class. Yeah.